Andrew V. Ogram. He is a professor at the University of Florida. Dr. Andrew Ogram received his PhD from the University of Tennessee and has been on the faculty of the Soil and Water Sciences Department at the University of Florida since 1996. He teaches several innovative courses in microbial ecology and the interactions between the environment and public health. His research is mainly focused on the Florida Everglades, the microbial ecology of the Florida Everglades. And today we have a unique opportunity to learn about this very interesting ecosystem located right in Florida, in our own backyard. We talk about the Florida Everglades with the students in context to several courses, including environmental policy and environmental sciences research. So let's give Dr. Andrew Ogram a warm welcome, and welcome again, Dr. Ogram.
Okay, thank you. I'm going to share my screen now, or at least that's my intent. Here we go. Oops. So Siddiqui, is my screen being shared now?
Yes.
Yes, I can see it. Mm-hmm.
Great. Well, thanks to Dr. Chauhan, my longtime friend and close collaborator for, as he said, it must have been over 20 years. It doesn't seem like it's been that long. I really appreciate the opportunity to talk to you all today. I've been fortunate enough to sit in on some of the seminars that you all have had this semester. It's been a great series, and so I'm really honored to be able to be part of that. Regarding questions, the audio so far has not been that good for me. I can't really hear very well. And so what I'd like to do is suggest that maybe you, rather than talk, if you put the things in the chat box or save your questions to the end, because I'm afraid I just won't hear you. If you can get my attention, then we can go ahead and take questions, but in progress. But it might be more efficient if we just do it at the end. So as Dr. Chauhan said, I'll be talking about the microbial ecology in the Everglades, and we've been studying that for quite a long time. Most of our work has been on the on methane production, and we're starting to get interested in the fundamental microbial ecology of mercury. And those are some things that I'll be talking about today. Let's see. So I'll give you a brief outline of who I am, a little bit about the Everglades, and then we'll go into the coupled cycles in the Everglades. Specifically, I'll talk about methane production, important greenhouse gas, as you know, nitrogen fixation, so the primary way that nitrogen, which is frequently a limiting nutrient or a limiting element in many ecosystems, is we think is brought into some of the peat of the Everglades. And then we'll talk about mercury cycling. And even though those three topics may seem kind of diverse and spread out, they're really all very, very closely coupled. These cycles are all coupled together, as most are. And then I'll go into a summary. So first, as Siddiqui and Dr. Chauhan mentioned, I work at the University of Florida. So the University of Florida, you may know, is just a couple of hours south of Tallahassee and Gainesville, Florida. Smaller city, but a nice one, I think. It's okay. And I work in the Soil and Water Sciences Department. This is our building, McCarty Hall. We have I believe it's 42 faculty members and probably over 100 graduate students. Our faculty are primarily located in that building, but McCarty Hall, but we're also spread out around the state in various research and education centers. The closest one to Tallahassee is in Quincy, and we have a couple of faculty members there, including a microbiologist, Sunny Liao. Our lab interests are on to study the effects of land use on microbial community composition and function. And that's important because microbial communities are really the fundamental entities that affect biogeochemical cycling. So biogeochemical cycling is this, the turnover and the transformation of elements that are critical to life, like nitrogen and phosphorus and carbon and sulfur, as we'll talk about. And so without the function of the microbial community, those elements wouldn't cycle. Everything cycles. And so we're really interested in the impacts of land use and anthropogenic impacts on the fundamental activities and composition of microbial communities and the relationship then to biogeochemical cycling that drives ecosystems. Most of our work is in wetlands. Most of it has been in the Everglades recently, and I'll go into that. And the primary players that I'll be talking about, I'll come back to this, but is this the guys that are in this this photograph that Dr. Chauhan took many, many years ago. It's the relationships between methanogenic archaea, or these prokaryotes that produce methane as a function of energy generation, and their cooperation, their essential cooperation with some sulfate reducing bacteria that don't really reduce sulfate, but feed them the electron donors or the energy that they need. And so I'll go back into that a little bit more later, more detail. So as I said, the work that we do is largely in the Everglades, and this is a map of the state of Florida. So as you know, Tallahassee is somewhere up here. Tallahassee is right there, if you can see my cursor, and Gainesville's here. And then what we generally think of as being the Everglades is a large marsh that's south of Lake Okeechobee. To do it justice, though, to be more proper, the greater Everglades ecosystem really starts with the Kissimmee River as it flows down through the state and into Lake Okeechobee. And as it moves south, it drains agricultural land and urban and natural lands, and ends up down in Lake Okeechobee. And as it drains those lands, it also, we also have septic tanks, we have agricultural runoff, we have urban runoff that's coming in there. So the Kissimmee River picks up nutrients as well as other pollutants as it runs down the state into Lake Okeechobee. And Okeechobee is pretty polluted, largely with phosphorus, but there are a lot of problems in there. Historically, the lake would spill its banks and run over into this part of the state as sheet flow. So that means it would just run off into this area that's flooded much of the time, but not all the time, and then go out as a very slow-moving river into Florida Bay. But that's not what I'm going to talk about. I'm going to restrict everything to this general area right here. So just to give you a little bit of introduction, much of the Everglades is a marsh. It's a complex landscape that you can see here. It's flooded much of the year, but it does dry out, and that's every year. Much of it dries out of the area that I'm going to talk about, and that's an important part of the ecology of the system. And it's composed, when it's not dry, it's composed of this area that's partially flooded, and then ridges here where you see, in this case, it's sawgrass. Historically, the Everglades was highly adapted to very, very low phosphorus concentrations. And so phosphorus is an essential macronutrient. What phosphorus? Phosphorus is important to the membranes in phospholipids that constitute, I think, all biological membranes in cells, whether you're a bacterium or a plant or an animal. And so this ecosystem was really, really well adapted to very, very low phosphorus concentrations. And what happened, unfortunately, is that, unfortunately for the Everglades, I guess, is that the area just south of Lake Okeechobee was drained for muck farms. So muck farms are just these farms that have high organic matter contents. And so you can think about what's happening. Sorry, I'm going to go back here. Over years, you have the accumulation of organic matter as the sawgrass or whatever, the different plants that are there would die, fall down into the below the surface because it's an anoxic system, and this is a complex issue. You have the accumulation of partially decomposed organic matter, and that's peat, and that's going to be much of our story. But it also is very valuable for growing some plants. And so as a result of that, those peat deposits, then it was viewed as being a very important source of muck farming. And so the Everglades were drained to form the Everglades agricultural area. This is kind of a top level view of the Everglades agricultural area. It's just below the lake. It's very flat, as you can see. It's drained to maintain the water levels down, and much of it is planted to sugarcane. There are a variety of crops that are planted in that area, but it's dominated by sugarcane. And here are sugarcane processing plants. It's fairly low nutrient, so phosphorus is put onto those fields as a fertilizer. And when the phosphorus, when it rains, or just as a natural course, the phosphorus runs off into the water, into these canals that you can see that drain the area. And so over the last 100 years, the area has drained phosphorus into this very, very low phosphorus, historically phosphorus region. And so it had the equivalent of fertilizing the northern Everglades. And so that really had some significant ecosystem impacts. Another thing that I'll be talking about later is that the pH, for various reasons, the pH is rather high. It's a little too high to grow sugarcane, and so the growers applied elemental sulfur to the soils. The elemental sulfur, the idea there was that sulfur oxidizing bacteria would convert that elemental sulfur into H2SO4, or sulfuric acid, and that would drop the pH a little bit. And so not only do you have sulfuric acid, you have sulfate then, and that sulfate also ran off into the Everglades, which was historically a fairly low sulfate area. And so what you have then is the fertilization of the Everglades. And I'll talk about that more in detail. It ran off into these areas that are just south of the Everglades agricultural area that are now called the water conservation areas. So these water conservation areas are set aside to, and specifically managed to provide water to the large populations here in West Palm Beach and Fort Lauderdale. Because of this phosphorus runoff, they also provide a great a great opportunity for ecologists to study the impacts of phosphorus fertilization on historically low phosphorus ecosystems because a gradient was set up, a gradient in phosphorus concentrations. And so here you can see that in the, in the northern area, right around where some of these canals were that emptied the phosphorus, you see very high concentrations of phosphorus as it enters. Then you see an area that's a little bit south of that high phosphorus concentration that's being converted into a kind of a eutrophied area, high phosphorus area, and we call that the transition zone. And then deeper into the marsh, it's relatively unimpacted. This is Water Conservation Area 2A. It's been intensely studied because of this gradient. What happens when you've converted it? What happens as you are converting it into a high phosphorus area, the transition zone, and how does that compare with the low phosphorus? And so this is a great outdoor experiment, really a large experiment on what happens. The most obvious thing that happens is you get a conversion of the ecosystem into different plants. You get a lot more biomass coming in because it's fertilized. And that sawgrass that we saw in the previous photograph is gone. It's outcompeted by cattails. So that's one clear indicator of high phosphorus. And so with this increased phosphorus, we get increased primary productivity or increased amounts of plant material, right? Now they can grow, they're not limited by phosphorus. Because you have more phosphorus and more biomass, more plant biomass, now you've got a lot more carbon, and that also stimulates the microbial communities that are there in the peat. And so you get increased levels of peat formation, rates of peat formation. You also get a lot more of these microbial activities that are going on, a lot more cycling, a lot more rapid cycling. And you get a shift from phosphorus limitation to nitrogen limitation. So phosphorus and nitrogen are the two primary limiters here. We also, Dr. Chauhan also saw that there's also evidence of carbon limitation in some areas of this area. So what we're primarily interested in is what happens down in the peat or in areas that don't have oxygen. So bacteria are very, very flexible. Many bacteria don't really need oxygen to breathe like we do. That's important for energy generation, but bacteria have figured out, unlike humans, how to use other substrates for breathing. So they don't really need oxygen. And that's where we're going to be focusing on now, are these anaerobic microorganisms. And so remember, we have this fertilization, and that leads to a lot more carbon coming in because we have a lot more plant biomass. What we're interested in is once it falls into the area below the water, what happens to that plant biomass? It enters what we can kind of conceptualize as being an anaerobic metabolic funnel. And so the plant biomass is composed of a lot of polymers like lignin, cellulose, hemicellulose, all of these different plant components. And the microorganisms depolymerize that. That's the first step to eating it. And then once they do that, they can kind of degrade or ferment the monomers that are there. And then it passes down into another set of organisms that mineralize it or convert it from organic carbon into inorganic carbon. And those inorganic chemicals are carbon dioxide, methane, and hydrogen. And so these are really three very different functional groups of microorganisms that participate in mineralization of plant material. We're really interested in what happens down here at the very bottom. What happens to these guys that are making the carbon dioxide, the methane, and the hydrogen? Okay, so we hypothesized that there would be significant differences in the microbial communities that are associated with a really high phosphorus area, because they've got a lot more carbon coming in, than there would be in the low phosphorus areas, uh, the more pristine areas. And so we use what's now kind of an old technique called a microarray that had, I think it's somewhere in the neighborhood of 30,000 genes that are primarily involved in the bacterial decontamination of contaminated sites. So it was really an environmental microbiology tool. And what we found is in the unimpacted area, there were 98 unique genes that are analyzed by this system, 148 unique genes in the impacted or the high phosphorus area. But in this transition zone between the two, we see 780 unique genes. So if we're thinking about the conversion of an ecosystem from a high contaminated area to a low contaminated area, generally you think that it's just kind of a mixing of the two communities right in the middle. But that's not at all what we have seen here. What we see is an explosion of catabolic diversity. Because, and we still really don't understand this, but in the transition zone, it's not just a mixture of the two. It's really a great expansion of catabolic diversity. And that suggests to us that there's increased carbon metabolism and metabolism and a greater amount of biomass in the transition area than there is in either of the end members. And we also see increased diversity and activity. And I think this is really interesting, intriguing, but we haven't been able to really follow up on that. So basically what you see is a very unique community right there in the transition zone. Okay, so going back then, what we're really interested in are these guys that have taken those activities that we just discussed, those unique genes. Most of those are carbon processing. They're part of the depolymerization and the degradation part of that anaerobic funnel, and they're feeding the byproducts down into the bottom, the mineralizers. And the most important mineralizers are pictured here in this photograph that Dr. Chauhan took 16 years ago. It's about, here you see different bacteria or different prokaryotes that are involved in the metabolism of butyrate. So butyric acid is just a fermentation product. It's one of those degradation products that I was talking about. These guys right here, these kind of short, rotund guys are centrophs. They're called centrophs because they work together with methanogenic archaea or methanogens. They will convert that butyrate into acetate. Then the acetate will feed these long prokaryotes, those are the methanogens, who will convert that acetate into methane. Also, they convert that butyrate into hydrogen. And a different set of prokaryotes, again, these long guys, different set though, will take that hydrogen and use it to reduce carbon dioxide to produce methane. So here we have this intersection of two different groups of methanogens in production of methane. And that's really what we're interested in. Something that's really interesting to note and important later is that these centrophic prokaryotes here, centrophic bacteria, are sulfate-reducing bacteria. So they are very, very different organisms from the methanogens. They use sulfate like we use oxygen. They use sulfate to breathe. And if they don't have enough sulfate, then they will work together with the methanogens. So it's kind of like a love-hate relationship. If they don't need the methanogens, they will compete. If they have enough sulfate, they will compete for nutrients with the methanogens and usually beat them. But if they run out of sulfate, then they will work together with the methanogens to make a living. And so as I just kind of alluded to, there are two basic groups of methanogens. Those that make acetate or take acetate and make methane, and then others that use hydrogen and make methane. And I'm not going to go into this, but basically what you think is that you get a lot more methane produced from acetate, which is one of those degradation products, than you do from hydrogen. But what we saw, or sorry, I'm getting ahead of myself. There are also two broad groups of sulfate reducing bacteria, or SRB, those that can use acetate and those that cannot use acetate. So right now you can see that there's kind of a competition that may be set up between the sulfate reducing bacteria and the methanogens for acetate. And they're all in this. We also have sulfate coming in to the Everglades from runoff that will feed the sulfate reducing bacteria to a certain extent. So what is that there's a selection for acetate oxidizing sulfate reducing bacteria in the impacted area. And that's what you would expect because we have sulfate coming in. And there is a selection for non-acetate using sulfate reducers in the unimpacted area. And basically what we see is just as we would have predicted, there's a competition for acetate between the sulfate reducers and the methanogens. So I'm not going to go into the methods here, but sulfate reducers outcompete the methanogens for the acetate in the high nutrient areas. And then down in the low nutrient area, we have a much more normal situation where there's kind of a balance. So the competition controls the basic physiological groups of methanogens and sulfate reducing bacteria that are in here. That's important because of not only because of the way that they make methane, but also because of some of the other things that these guys do. All of these cycles are coupled. So not just methane, not just the acetate, but also, as I'll discuss, nitrogen cycling, which is important to the continued growth of these organisms and their activities, but also to mercury cycling, as we'll get to in a minute. So something that kind of surprised us is that methanogenic bacteria fix nitrogen. That's been known since the early 80s, but there's been virtually no work that's been done until we did it in a natural environment. There'd been some work done in fermenters and in digesters in the laboratory, but not in out in a real environment. And so what we found is virtually all of the methanogens are able to fix nitrogen, and that almost all of those nitrogen fixers are fixing it using hydrogen. So remember, there are these two different groups, the acetotrophic and the hydrogenotrophic methanogens. And the hydrogen users are the ones that's fixing the nitrogen. Remember that we've seen a selection along that gradient for hydrogen fixers. So what does that mean? Well, it means that if there's a balance between nitrogen fixers down in the peat, it's probably between sulfate reducing bacteria and the methanogens. In the nutrient impacted area, where we see more methanogens, we see a greater amount of nitrogen being fixed by the methanogens. So not only are they important in a number of things like hydrogen control, but also methane production, they're also the guys that are fixing the nitrogen because of this competition. As we go down the gradient, then we see this dark blue is representing the sulfate reducing bacteria, and there's a complete shift there. What we see is that the sulfate reducers in the low nutrient area are more responsible for nitrogen fixation down in the peat. So this is important because nitrogen is limiting down there, right? These bacteria need nitrogen, or prokaryotes need nitrogen in order to make proteins, in order to grow, in order to make the enzymes that are essential to methane production or whatever it is that they grow. And so the processes that control nitrogen fixation are really important. So it's not just the guys that do it, it's the processes that drive this nitrogen fixation. Okay, so I'd like to summarize this part right now. I think I may end up early. In the nutrient impacted peat versus the low nutrient impacted peat, there's a shift in primary productivity from phosphorus limited to nitrogen limited. And when we put in phosphorus, we change everything from the plant type, the plant density, to the microbial communities that are responsible for cycling everything. As we have a front that is moving down, and this phosphorus moves down from the north down to the south, just with water flow, but it's it's slowly, very slowly. You get the conversion of some of this kind of pristine area that's phosphorus limited into this wholly other thing. It's you see this expansion of catabolic diversity, and so you see really rapid carbon cycling, methane production. A lot of things are just really ramped up, not just because you have more bacteria, but because there's a much greater diversity of microorganisms that's present in that transition zone. And then we go down to the oligotrophic area. We have something that's really quite different too. Those are the guys that are really well adapted to low phosphorus and low sulfate. That impacts the relationship between sulfate-reducing bacteria and thanogens, the way that methane is produced, and the way that nitrogen is fixed along that gradient. Okay, so if this affects the way that methane is produced and the way that nitrogen is produced or is fixed, then are there other pathways that may be impacted? And it turns out, we think, that probably that it does affect the way that these other cycles. And so that one of the things that we got interested in, I think a lot of people are interested in, is mercury cycling. So this is from 2016, but probably the map for 2020 doesn't look that different. This is a map of mercury deposition. So mercury deposits from the atmosphere down to the surface through just kind of falling, but primarily through rain. And what you'll see is that Florida is red, and what you can't see is that southern Florida, where the Everglades is, has some extremely high rates of mercury deposition. The reason for this is because we have air currents that are bringing mercury from Asia, China and India, across the earth. And they are, because of the convergence of high rainfall and some of these air currents that are global, you have mercury being brought down into Florida and the Caribbean at very high rates. Okay, it's brought down, as I'll discuss in a minute, it's brought down as primarily as Hg2+, inorganic mercury. Because of the Everglades being a site of very high mercury deposition, there is a lot of concern that mercury, a very, very potent neurotoxin, may be affecting public health as well as wildlife health. This is a picture of an ibis, and a study conducted here by some colleagues at the University of Florida have shown that mercury will bioaccumulate through small organisms up through fish up to top level predators like wading birds, and accumulate in their tissue and impact mating habits. So we know that there are some fairly significant impacts on wading bird health because of the mercury. We're also concerned that there may be potentially human health impacts of mercury. So mercury is a potent neurotoxin. It impacts wading bird reproductive mating habits, but which will decrease their numbers. But also it is a potent neurotoxin, and it's been shown in numerous studies to decrease human IQ. So it'll decrease your IQ by depending on the concentrations of the mercury, down a few points if the mother ingests mercury, such as eating predator fish like bass here. So these signs are posted around the Everglades. Don't eat the fish because of mercury. This is what the cycle of mercury looks like. It is largely arises from fossil fuel production. And again, it's not so much in the United States that we see. It's from Asia, from China and India, primarily, I think from China. And then it rains down into the Everglades. And if it rains on dry land, it's not such a great concern. But if it comes down into an area that has anaerobic zones, anoxic zones, then anaerobic bacteria or prokaryotes will convert that inorganic mercury into methyl mercury. It'll put a methyl group on it. And that is what is bioaccumulating. That's the chemical that's of greatest concern. So we have inorganic mercury being converted by bacteria, anaerobic bacteria to organic mercury, methyl mercury, and that will be taken up eventually by fish and then eaten by birds and potentially by humans. And that's where the great concern is. Okay, as I had said before, there's a gradient in sulfate. And so you have, because of the Everglades agricultural area probably, you have a higher concentration of sulfate in the northern areas of the Everglades than you do in the south. And so that gives us a really a great experimental setup to study the impacts of sulfate and phosphorus production, organic carbon coming into a system on mercury transformations. And so we've conducted some analyses up in the areas that are very close to the Everglades agricultural area, where there are high concentrations of sulfate, relatively high, not like seawater or anything like that, but relatively high concentrations, down into this area and water conservation area 3A that has very low sulfate concentrations. I should say that sulfate reducing bacteria, who we're interested in, are thought to be the primary metabolism that controls mercury methylation. So if you have high sulfate, then that turns those guys on, and it will convert that mercury into methyl mercury. And so that's a real serious concern. But what happens down in the other areas? Okay, if we look at the distribution of genes that are implicated, bacterial genes, prokaryotic genes that are implicated in methylation, down this gradient, what we see is that we're going down the gradient now. At high concentrations of sulfate, we see this area, this light blue area, this represents the distribution of the genes between the sulfate reducers, methanogens, firmicutes, which are fermenters, and some other groups of bacteria, really diverse groups that are capable of methylating mercury, but by far the dominant group is this light blue, and those are the sulfate reducing bacteria. They also are those centropes. So we don't know if those organisms are functioning centrophically when they methylate mercury, or if they are functioning as sulfate-reducing bacteria, probably they are functioning as sulfate-reducing bacteria. And we go a little bit further down the gradient, and we see, again, this light blue is the sulfate-reducing bacteria. They're the dominant group that's responsible for mercury methylation, we think. But then we get down a little bit further south where we're don't have very much of that sulfate at all that's coming from the agricultural area. And the sulfate-reducing bacteria, the light blue area, is very small, and we see an increase in the amount of methanogens that are responsible. So again, it's our two favorite groups here that are responsible, and they shift along that gradient. This is important because if you run out of sulfate, you probably are still getting at least a small amount, if not a significant amount, of mercury methylation down in those areas. The Everglades is a complex system. It does not just have peat, and I've only been talking about the peat, but it has flock. And if you go above the peat, there's this unconsolidated area that has partially decomposed plant material and a lot of carbohydrates and stuff that are produced by microorganisms. It's kind of a yucky type fraction. Then you have water, and then you also have periphyton, and the periphyton can float on the top of the water if it's a mat, and it can also live on the surface of plants. And so we were interested in what happens as you go from the soil up to the flock, up to the periphyton. And so, and this periphyton is stuck on the plants. So what we see here is the composition of the bacteria that can methylate mercury down in the soil and down in the peat. Oh, sorry. And this is in an area that has sulfate in it and an area that does not have sulfate. So we're just comparing two sites here. And again, down in the peat, we see sulfate-reducing bacteria are primarily the primarily dominant organisms that are responsible for this. As we move up into the flock, the story becomes much more complex. We see methanogens, which are these guys, and firmicutes, which are the light blue. Those are fermenters, and then a little bit of the prokaryotes, or the sulfate-reducing bacteria. Then we go up into the periphyton. The sulfate reducers become important again, and then methanogens and firmicutes round it out. As we go down in sulfate concentration, again, we have really a substantial number of these sulfate reducers down in the peat. But if we go up into the flock, then we see a lot of methanogens, a relatively small amount of the sulfate reducers, and then some of these other metabolisms. And the same is true for the periphyton. We see a lot of methanogens and a relatively small proportion of the sulfate reducing bacteria. So this is important because we wonder what the food is for these fish that are eating it. So the fish probably aren't down here in the mud in the peat eating the peat. They're up eating the flock probably, or the periphyton, eating something in the flock or the periphyton. And so that means that these metabolisms, in addition to the sulfate reducing bacteria, may be more important than just the sulfate reducers. So we thought, well, we have these data. I'm not going to go into this graph very much, as to go into this would take quite a bit of time that we may not have. But we have really quite a lot more data than I've showed you. But we thought, well, we have all of this data regarding the distribution of the different types of genes, the concentration of genes that are involved in mercury methylation, and we have methyl mercury concentrations. Can we correlate the concentration of genes with the concentration of methylmercury that's present in the system? And the answer is no. That's really what this is telling us. These are various genes involved in mercury methylation, sulfate-reducing bacteria genes, methanogenic genes. There's all kinds of stuff in here. Everybody likes carbon is really what this is saying. But this is methylmercury. That's an outlier. So the numbers of these genes that control the concentrations don't seem to have any relationship to the numbers or to the amount of methylmercury. And so that's the bottom line is really what's controlling mercury methylation, if it's not just the genes. And again, this is a complex slide, but the bottom line here is that we think that its bioavailability is the nature of the dissolved organic carbon can significantly impact the amount of mercury that enters the cell. And so that's something that we're looking at now, is the relationship between bioavailability of mercury and entry into these cells that have the mercury methylating genes. And so we hope that that will give us some insight into what's controlling the methylation rates, because it's clearly not just the bacteria. One of the things that we're really interested in is what's happening in the plants and around the plants. And that's what this is showing us. We think that probably plants are important in bioavailability, and we think that probably also the something that this slide is showing you, you could see MEHG, methylmercury, going to HG. This is a demethylation step in all of these. The red is the HG. So remember, methylmercury is the critical component for that's primarily the toxin that we're interested in, that bioaccumulates. Bacteria, many of the bacteria that I've just talked about, not only will methylate mercury, but they will also demethylate mercury. And that's something that is, we know very, very little about. There have been some studies that have shown that sulfate-reducing bacteria and our friends the methanogens also demethylate. So not only do they methylate, they demethylate. So it's a very, very complex system. So in summary, then nutrient pollution changed the fundamental ecology of the Everglades, including the pathways toward methane production. It also changes the processes that control nitrogen fixation and the processes that control mercury cycling. And we have a lot of work to do to truly understand the fundamental microbial ecology of mercury cycling in these systems. So I don't want to go into all of this, but a lot of people contributed to this, including Dr. Chauhan, who really laid the groundwork for a lot of what I've talked about with his fundamental work on centros and methanogens. So with that, I say thank you. And if you have any questions, I'll try to answer those.
Thanks. Yes, there's a lot of questions, actually. So first of all, thank you for that. Really appreciate it. And, just can't thank you enough. The first question comes from Ms. Ivory Council, one of our PhD students. What types of tools or indicators are used to determine exactly where the transition zone is located?
Okay. Yeah, that's a good question. So this is, it's kind of a moving zone. I'll say that first, because the front of phosphorus is moving. So it's like a moving front. The tools are that you can see that there are mixed zones of cattail, which is more characteristic of the high phosphorus area, and and sawgrass. So there's this mixture. We also, you can see in the laboratory, you can bring these samples from the peat back, and you see really high rates of methane production and carbon dioxide production in those areas. And then you also, there's also some interesting work that's been done to look at the enzyme assays there. So it's like the organisms, the microorganisms that are in the phosphorus limited areas are phosphorus limited, and their enzymes that they produce reflect that. In the area that is not phosphorus limited, they are nitrogen limited, and their enzymes kind of reflect that. The ones in the transition zone are neither here nor there. It's really quite interesting. They're kind of a mixture of the two, but they're not really limited in phosphorus or nitrogen. I suspect that they're limited in carbon at that point. And that's why you get this great expansion of catabolic diversity, genetic diversity.
Cool. All right. Second questions from DeMarcus Robinson. Did you also look at iron reduction, oxidation that could also contribute that the absorption and release of phosphorus? And then he had a comment of, I think that gives another relationship on how phosphorus is changed.
Yeah, that's a good question. Phosphorus is, iron is, there's not a lot of iron in these areas of the Everglades that we looked at. There is some, but it's not, we don't think it's enough to affect the microbiology of the system. And as far as iron and phosphorus, that's not what's controlling the phosphorus availability. It's calcium. So I didn't talk about that, but this is underlain, like much of Florida, by limestone, by the remnants of coral reefs, which is calcium carbonate or limestone. And so these are fairly high calcium systems. And so the calcium and the phosphate form to produce hydroxyapatite, and that's insoluble. And so that controls the availability, not the iron.
Okay. Next question is from Christy Crandall. As the nutrient input shifts in amount and concentration so that the nutrient gradient actually shifts in location, will the system maintain the diversity, or will the composition diversity shift with the gradient? She's wondering if the system returns to previous conditions if Lake Okeechobee, Kissimmee River is cleaned up eventually.
That's a good question. That's a question that a lot of people have been asking too. The answer is you can see the microbial communities and their functions really changing as the front comes through. The sugar companies in the Everglades agricultural area have really made great strides in stopping phosphorus from coming down. And so they've really cleaned up their act quite a lot. And so the phosphorus that's being put into this system is much, much lower than it was 10, 20 years ago. What's happening now, though, is that everything is redistributing. So the phosphorus that's there in the high phosphorus area is slowly being released from the organic phosphorus, and it's moving south. And so it will be a very, very long time before all that phosphorus washes out. And the same would be true for what's happening in Lake Okeechobee. It's just going to be a long, long time for it to naturally cleanse like that, just because there's so much phosphorus that's in there.
Okay, next question comes from Dr. Pathak. What options do we have to decrease the delivery of atmospheric mercury to the Everglades? And what changes we can do to reduce the production of methyl mercury? Like what environmental conditions can be brought for less favorable conditions for methyl mercury?
Well, one of the hot areas of I don't know, debate or litigation maybe even, is that sulfate should not be used, or sulfur should not be applied to those fields, or it should be intercepted before it goes into the Everglades, because that's what's driving much of the methylation. Definitely not all, but at least the significant amounts appear to be sulfate driven. And so if we can stop the sulfate from coming in, then that'll be great. The problem with the sulfate is that it's also a little bit like, not exactly, but it's a little bit also like the phosphorus, in that there's still quite a load of sulfate in these northern areas that can be redistributed south. Phosphorus is a little, or sulfate is a little different, because sulfate reducing bacteria produce hydrogen sulfide, and some of that can be volatilized out, and so you can blow some of it off. The ultimate thing, it would be to stop the source, and the source for us anyway in Florida is Asia. And I believe that India is making great strides to reduce the amount of coal production or coal-fired power plants, and I'm sure China is too. And so I think they're making great efforts their efforts to convert their power generation to more cleaner technologies is really going to be the main thing.
Okay, and then Christy's back. Did you measure these samples under different flowing conditions? Is there flow, therefore more oxygen diffusion in flowing areas?
That's a good question. No. No to both of those questions. There's no, the areas that we're studying are, in the peat, there's not much diffusion of oxygen. What happens in the peat is you've got this floc layer in much of the area, and the bacteria that are in the floc consume the oxygen really rapidly, and so very little oxygen gets down into the peat. If it comes in, it's not because of flow, it's because of plants. So plants, some plants have the ability to transport oxygen down into the root system. And so you get a small area around the roots that's aerobic, but it also doesn't diffuse out very far because of all of these bacteria that are eagerly using that oxygen to breathe. So most of it is not affected by flow. The periphyton that may be floating has very, very interesting dynamics as far as oxygen is concerned. It can be during the day, it can be highly aerobic because of the different bacteria that are in there that are photosynthesizing, and in the process they're producing oxygen. But at night, when there's no photosynthesis going on, there's no oxygen production in the interior, and so the redox potentials or the oxygen concentrations can go down very low. But I don't believe that flow in those compartments is important. It may be much more important in the water column, but not so much in the ones that we're studying. So that was also a good question. Thanks.
All right, and then Mr. Robinson, he has a comment. I don't know if you want to add to it or anything, but he just says, I think it would be interesting to see the rates of consumption of carbon, nitrogen, sulfate, and mercury using stable or radioisotopes and comparing it with the gene data.
Yes, yeah. I didn't show you a lot of data that we have, but we and other people have done quite a bit of that. We have done some work in the laboratory with stable isotopes of mercury, and consequently also sulfate reduction and methane production. Much more detailed experiments we have, and then a lot of other people have also done similar work. So that's a good point.
Okay, and then this is from Dr. Manashi. I know, I'm so sorry. I know I messed that up a little bit, but she's saying great presentation, thanks. I'm unclear how the higher methyl mercury, I guess saying consumption does not correlate with methylation genes.
Yeah, that was kind of disappointing to us, but nothing is easy. So a little bit of background on those methylation genes. they methylate mercury gratuitously. So that means that they're genes that encode enzymes that will transfer a methyl group from one substrate onto another. And they just happen to be able to transfer a methyl group from some substrate onto mercury. It's part of their carbon processing, their carbon metabolism. They don't care about the mercury. It's just kind of, the mercury comes in and it gets kind of put into that pipeline and it gets methylated. And so they're not turned on by the presence of mercury. They're just doing their thing. And so many of these organisms have the ability to methylate mercury if the mercury is there. But it's independent of whether even there's mercury there. We also don't know what controls their expression or the production of the proteins that do that in the environment. Very little is known about this. And we have looked at messenger RNA that would tell us the rate of activity of those genes in the environment, but the concentrations are so low that of the mRNA that we can't really measure it. So, that's why, I guess, we don't know if those genes are turned on. We don't know what turns them on. We don't know anything else really. What we believe, though, is that the bioavailability of the mercury is really critical in these systems. And that's because there's a lot of dissolved organic carbon in all of these environmental compartments. And depending on the nature of the dissolved organic carbon, it may bind the mercury and facilitate its movement into the cell. And then it's going to get methylated. We also don't know much about the mercury demethylation rates, and that's something that was investigated in the 90s and really should be picked up again. That probably is also an important aspect of this disconnect between the methyl mercury concentrations and the gene concentrations.
Okay, great. And I know we're running a little bit over time, but we have two more questions. And guys, thank you all so much for participating in this. And it's just been phenomenal. Just as someone who studied business and economics, you guys have really forced me to, I'm just taking all my notes and, all right, what's this exactly and how does this work? So just phenomenal on both ends, presenters, Dr. Chauhan and students as well. But Mr. Robinson's back in it. Again, he's asking, is there high rates of sulfate reduction, or do you see concentrations in sulfide in these environments?
Yeah, well, high rates is kind of a kind of qualified. We see significant rates of sulfate reduction in the sulfate impacted areas in the north. It's not as high as you find in estuaries, for example, because the sulfate is much lower. But we do see sulfate reduction there. One of the aspects of sulfate reduction is it will produce sulfide, HS minus, and that sulfide will complex the mercury, and it can produce insoluble complexes of mercury sulfide that just kind of precipitate out. So, and that's also been observed in some of these really relatively high sulfate concentration areas. So hydrogen sulfide will bond with mercury and make it unavailable.
Okay, and then from Dr. Washote, who's also he's our director of our core labs. Are you aware of any other areas? Oh, excuse me, I lost the question as you exited out there. Are you aware of any other areas in Florida with elevated mercury concentrations compared to the Everglades?
No, I'm not, and that's a good question because we've been looking for another site just recently that we could compare with the Everglades. There probably are. There are other marshes. You know, the thing that you would look for is something like the Everglades that has a large surface area that's flooded, and be down in the south where we have more rainfall and the convergence of these air currents. So we're curious about Blue Cypress Marsh. That's the headwaters of the St. Johns River. That's north of the Everglades, but we don't know. They don't know, so don't know. That's the answer to that. I think probably there are, but we don't know. I don't know anyway. Maybe that's the best answer. I don't know.
I think we had a really good session. I have just one last question. I don't think anyone else posed any questions, right, Mr. Perry?
No, that's it for now.
Okay. I just had a quick question. I'm kind of curious on, you know, a great presentation, great set of data, and thank you very much for that, Dr. Ogram. I was wondering if there are plans to develop bioindicators. You know, you do see many of these organisms responding to certain environmental conditions. So are there any plans to develop them as bioindicators and then perhaps link it to more geospatial tools, you know, development of apps and geospatial apps that that decision makers can use to figure out whether there's phosphorus pollution and other kinds of pollutions in other environments?
Yeah, that's a real good question, Ashwini. As you may not remember this, but that's really what got us into this, was to assess some aspect of microbial community structure for as being an indicator, a quick indicator of pollution. And we've kind of gotten away from that, and I don't know if there are others. One of the things that we're interested in is working with landscape ecologists and GIS specialists to kind of try and transfer this up to the landscape scale, some of these data, but we haven't been funded on it. So, you know, we've gotten a lot of rejections, but we haven't gotten it funded.
Right.
But I think it's certainly plausible.
I just wanted to mention that we have a geospatial center funded by the restoration grant. It's called Caesar, and Dean Abanusi leads the Caesar Center here in FAMU. So perhaps we could all explore joint collaborations. And Christy is a GIS, has GIS background. Christy, can you speak a little bit on Caesar?
Yes, my project actually uses some really high-level geospatial data. It's too bad Joe St. Peter isn't on this call, but he's doing amazing things, identifying vegetation at the 1 meter by 1 meter resolution. And right now, my project is covering the Apalachicola region. But it, you know, this that data is available. It's from satellite imagery, the national. It's called NAPE. I forget what the acronym stands for, but the spatial vegetation identification is resolution is so incredibly high that within a 10 meter by 10 meter grid cell, you can identify what the percentage pine is. For example, I was just thinking, you could identify what the percentage of cattail was, or, you know, I don't know what else you could use. There are some variables about particulate, you know, the color of the water using the palm, suspended palm coloration of the water to relate that. But anyway, I'm sure there's a ton of things that could be done, and we have the tools and capability in our center to do it.
Yeah, that would be really, really interesting. Maybe we should talk about that.
Yes, definitely. talk to Dr. Ibanesi.
Yeah, so maybe we can connect, or Dr. Sean, you can connect Dr. Ogram with the CSER team and then you guys can meet and properly or fully discuss plans to better collaborate. I think that would be great. I know we're past time, and I know several of you have to go on to other things. So if there's no questions or anything, I will make a quick brief announcement. We are having our final session next week on Friday, the same time, same place.